You know, for generations, certain words in medicine, well, they felt like a final verdict. Genetic blindness was definitely one of them. But in this explainer, we're going to dive into how a full-blown scientific revolution is turning that verdict completely on its head.
Just think about that question for a second, because it's not hypothetical anymore. For thousands and thousands of people, the answer to that is changing, like right now, in real time. We're talking about the very real possibility of stopping and, yeah, even reversing a condition that was once considered just a one-way path into darkness.
Okay, so to really get how huge these breakthroughs are, we first have to understand the opponent. And at the heart of this whole story is a disease called retinitis pigmentosa, or just RP for short.
Now, what's so important to get about RP is that it's not just one disease. It's more of a clinical syndrome, this whole collection of conditions that can be caused by faults in over 270 different genes. Can you imagine trying to fix a machine that has 270 different ways it can break? That's the insane challenge scientists have been up against for decades.
The way RP progresses it's like this tragic but predictable set of dominoes. It all starts with one faulty gene. That one tiny error causes the rod cells, the ones we use for night vision and seeing things on the periphery, to die off. But once they're gone, the area in the retina actually becomes toxic. And that toxic environment, in turn, starves and kills the cone cells, the ones we need for sharp, detailed color vision. And this predictable chain reaction is exactly what scientists are now learning how to interrupt.
For the longest time, the only thing doctors could do was just manage the decline. Things like vitamin supplements, special visual aids. They could offer a little bit of support, sure, but they were basically just sandbags against the rising flood. They couldn't do a thing about the root cause. But oh boy, that is all changing.
And this brings us to the first revolutionary tool in the new arsenal. It's also the most direct approach you can imagine, going straight for the broken gene itself. You can think of it as a form of molecular surgery. So instead of just treating symptoms, the whole goal of gene therapy is to fix the actual blueprint. It's designed to give the eye cells a new working copy of their instructions, which basically stops that very first domino from ever falling.
And this is just brilliant. There are two main ways to perform this surgery. On one hand, you've got gene replacement. This is where a harmless, re-engineered virus, called an AAV, acts like a microscopic little delivery truck. It carries a correct copy of a gene right into the retinal cells. That's the idea behind a therapy called laru-zova. But what if the gene is too big for the delivery truck? That's where gene repair comes in. A special molecule called an ASO, which they use in a therapy like sepofarsen, it doesn't replace the gene. Instead, it intercepts the faulty message coming from the gene and corrects the typo on the spot, so the cell can make the right protein. So cool.
And look, this isn't just theory. The results are real and they're happening now. A therapy called AGTC-501 has improved patients' vision and, get this, held those gains for at least 2 years. Laru-zova is so promising it's already in the final stages of clinical trials, and sepofarsen is blowing the doors open to treating mutations that we once thought were totally unreachable. This is happening.
Okay, but this brings up a really critical question, right? What happens if the dominoes have already fallen? What if those essential light-sensing cells are, well, already gone? Gene therapy, after all, needs a living cell to work on.
And that's exactly where our next tool comes in. If gene therapy is that molecular scalpel, you can think of cell therapy as a biological patch. The goal here isn't to fix the cells that are left, it's to actually replace the ones that have been lost.
So let's look at this groundbreaking example, OpCT-001. It's incredible. Scientists can actually take a patient's own cells, say from their skin, turn the clock back on them to become these super powerful induced pluripotent stem cells, or iPSCs, and then guide them to become brand new, healthy light-sensing cells. Then they transplant those into the eye. And here's the huge advantage. It doesn't matter one bit which of those 270 genes caused the problem in the first place, because you're simply bringing in a fresh workforce.
And what's absolutely amazing is that these new cells, they do more than just see. Recent studies have found that they also release all these protective proteins. So they don't just patch the hole, they actually create a healthier environment that helps protect the remaining struggling cells from dying off. It's like a 2-for-1 benefit nobody saw coming.
So we can fix genes and we can replace cells. But what about the most advanced cases? The ones where the retina's light-sensing hardware is almost completely gone. For that, we need a solution that's even more ingenious. Welcome to the world of optogenetics.
This approach it's a complete workaround. It just bypasses the dead photoreceptor cells altogether. Instead, it uses gene therapy to insert a special light-sensitive protein into the next layer of retinal cells, cells that normally just pass along signals but don't see light themselves. It essentially turns them into a brand new set of light sensors. It's literally a biological rewiring of the system.
20 days. What on earth could be so important about 20 days?
That's how quickly a next-generation optogenetic therapy called ZM02 sailed through the FDA's initial review process to start clinical trials. I mean, a 20-day turnaround is practically unheard of. It signals this incredible confidence in the tech. And why? Because this new version is so sensitive to light that patients might actually be able to see in normal, everyday light. No bulky amplifying goggles needed like with the earlier versions. It's a total quantum leap forward.
So, let's just take a step back and look at the big picture here. We don't have one single magic bullet. What we have now is something way, way more powerful, a versatile and growing toolbox.
This toolbox right here, this is what the new era of sight looks like. You can see so clearly how each strategy fits a specific need. You've got gene therapy to fix the source code early on. If cells are already gone, cell therapy can come in and rebuild. For the absolute most advanced cases, Optogenetics offers that brilliant rewire. And then, supporting all of them, you have small molecules that help protect any cells that are left. The big takeaway is this: there is now a tool for pretty much every single scenario.
Now, of course, the work isn't done. I mean, there are still some really critical hurdles to clear. Scientists are fine-tuning how to manage the body's immune response, they're figuring out how to pinpoint that perfect treatment window for every single patient, and they're working to make sure these therapies can safely restore a full field of vision by treating both eyes.
But the fundamental truth, the whole paradigm has shifted. The very idea of an untreatable genetic blinding disease, well, that's fast becoming a chapter in a history book. The story that's being written right now, today, is no longer about just managing a slow decline. It's about repair. It's about recovery. And for a growing number of people, it is about the return of sight.